Okay. Hi, everyone. Today we're just going to spend about 20-25 minutes talking about our EEG analysis process, going through a sample of what we now call brain functional analysis. Other people call it quantitative EEG or QEEG. And our techniques are somewhat unique, so we're just going to talk about kind of the process we go through, and then we're going to go through an example of someone's BFA, or brain functional analysis, and what that means and what we think that means for that individual. Okay? So essentially what happens in the pipeline of the EEG processing is obviously the EEG is collected. We are now in our clinic using a Neuroscan Synapse 2 amplifier with either 32 or 64 channels of EEG. Once that data is processed and forwarded over to us, it is then run through a pipeline where we use EEG Lab and other software components, and we do what's called an independent components analysis. So independent components analysis is a statistical procedure where you can isolate statistically unique portions of an EEG and separate them away from each other. And we'll talk about what that means in a second. But essentially, you can take one segment that is, say, muscle artifact, and separate that out from another segment that, say, is real alpha activity in the EEG. And you can then delete the muscle artifact and just keep the EEG. So it's an excellent way of getting a cleaner EEG, and it's also a very good way of isolating different components within the EEG itself. I think I have kind of an example down here. So for example, here you can see, sorry, let me get close to the screen here, but this top one is an EEG eye movement, a blink essentially. And here's a similar one here. This would be what's called muscle activity. And these in here are different components of real EEG that we want to keep, but they're different from each other. So one might be theta activity, one might be alpha activity, one might be activity over the right parietal lobe or the right frontal lobe. And we can keep and isolate all of those from each other and then include them in the analysis more specifically. So it's a way for us to eliminate or reduce artifact as much as possible, and also then to separate out the EEG into different components that can be analyzed uniquely. From the remaining components, we do what's called a dipole source localization, where we get the source or region in the brain deeper than the surface where that EEG is coming from. We then take all of those dipoles and enter it into what's called multivariate Granger causality analysis, which is a sophisticated statistical measure of connectivity, and specifically effective connectivity, which tells us that what happened at point A is actually causing what happened at point B, and vice versa. And then all of that is entered into what are called graph theory metrics, which are metrics of regional connectivity that you can look at across different locations. And then the last step we follow is we enter all of this into a normative database. This is the QEEG part that most people also do, where we compare absolute and relative power to a normative database to determine if someone is off from the norm, if these are abnormalities, and then we can source localize where those are coming from in the brain. Okay. So now we're going to look at someone's example here, see if we can help to understand this and their situation. So this patient came in to see us with difficulties with memory, confusion, suspiciousness of other people, and anxiety. She was actually hospitalized at an inpatient level for a couple of weeks and went to their day program for 2 weeks thereafter. Didn't notice any improvement, and then was referred here for a more comprehensive evaluation. Oops. So what we do is we go through their EEG data collection. This is a 32-channel EEG, so you can see the 32 channels displayed here. Eyes closed and eyes open. And you'll notice, for example, this is the spectral plot of the EEG that shows the frequency across the bottom from 0 to 50 hertz and power spectral density on the side, which is essentially amplitude or the size of the waves. And it shows us peaks and valleys in alpha, beta, and higher beta as well. This is before we applied ICA for artifacting, and this is what happens afterwards. So you can see there's a sizable difference in activity you see in the very low ends and the higher ends, which are actually artifact. So it changes our EEG, and now we look at more the pure EEG itself. We then, let me see, what's a better one here? Oops. So once we take out all the artifact, we have 4 primary, what are called independent components that we're looking at. And before we do that, we'll just kind of focus in a little bit on what we see here. One of the things you'll notice here is the peaks and valleys. So there's a peak at alpha, which oftentimes is normal with your eyes closed, but we have 2 secondary peaks in beta frequency bands at about 15 to 17 and 20 to 24 hertz. And these maps now are showing us the activity in these different regions. So at 10 hertz, you see sort of red areas over the right and left side in actually what's called a mu rhythm pattern, which is a distorted form of alpha that affects how you think, how you relate to other people, how you modulate emotions, things like that. And then we can see an excess of beta activity more over the right side, which frequently, again, has a lot to do with anxiety. As we go down to the components, we can now see that they further isolate those regions of the brain. So we have 2 on the right side, 1 over the central and 1 over the parietal lobes, a central location and a left central and parietal location. And each of these represent different type of activity in the EEG. And we can then take the dipole localization of where those are coming from in the brain. And even though we saw the activity that one of them was in the back of the brain, the source of it is all over the middle central part of the brain. Those are areas actually deeper in the brain into the temporal lobes and the limbic system. And what do those control? Well, they're most important for controlling emotional regulation, social interaction, and memory functions. And that's essentially what she's coming to us for. And then again, we can look at each one of these individual frequencies, components, and their activity. And then we skip down to what we call the Granger causality. So this is our connectivity analysis. That's very important to understand how to interpret this. So one of the things you look at is a scale on the side here that goes from 0 to 10. So that means that all of these circles and lines represent a connectivity from 0 to 10. The possible scale can go from 0 to 100. For an adult, you would want it to be between 25 and 40. Okay? So that means anything above 40 is very high connectivity or too much, and anything below 25 is too low connectivity. So her highest value is a 10, which means she never has any value during the CEG that goes above a 10, or only about 40% of what it should be for a normal functioning adult. Okay? The red circles and lines represent higher values, so closer to 10, and the blue lines, the dark blue lines, represent lower values, which are down around a 3 or a 4. So when we think of a 4, and we compare that to the norm that should be a 25, we're talking about someone that has probably 80% lower connectivity than he should have, which is very, very significant. These are taken across time. So this is how it changes from 0 seconds to 88 seconds to 177 and so on. And you can see in the middle part of the brain, there's this attempt to try to increase connectivity, but it never really gets to the sides or to deeper levels to help her perform that function. From that, we go to another measurement called reciprocal causality. What this does is it takes our measurement of connectivity and it says, okay, let's get this in the middle here. If what we have from location 1 to 4 is a certain value, and from 4 to 1 is another value, let's subtract them and determine which one is causing or pushing it more in the direction of the other one. In a fully functioning brain, these values should be relatively equal. There should be a balance of excitation or input from one part and inhibition or regulation from another part of the brain, and they should go back and forth. When there's an imbalance in that, we have problems. So in this picture, you'll see there's arrows pointing in certain directions. Like there's 2 pointing to this one in the right parietal lobes there. One up in the frontal lobe, one over to this right temporal region. And those become sort of weaker areas in the regulation of activity. So for her, we see most of that over on that right side around parietal, central, temporal, and frontal regions. And if you go back up to here, you'll remember we saw the majority of this excess activity over that right side of the brain as well. So the idea is that the connectivity imbalance is causing the brain to fire in ways it shouldn't. And that causes excesses of what we typically call power or amplitude or the waves of the EEG activity. And that's what leads to their symptoms. From all of that, we go to something called graph theory metrics, which then is a statistical way of grouping all of those measures of connectivity into one statistical measurement. Okay? So the one we look at the most is what's called path length. Hers is a 34. Path length is the average efficiency of the connections across all possible connections that we've looked at. A healthy path length would be under 20 or so. Okay? So that tells us that hers is higher or less efficient than it should be. We then break that up into radius and diameter. Radius is a 31. Radius is the efficiency of the shortest connections. And diameter is the efficiency of the longest connections. So that tells us that her diameter is much worse than her radius. So we would want to go back here and look at this and begin to understand that it's not just the short connections that are important, but the longest ones across that are. So when we think about setting up a neurofeedback protocol, we want to make sure to pay attention to that, to incorporate all of that information. And then our last step is to take all of that and compare it to a normative database for what we call absolute power and relative power. So here are the values for absolute power. These are the raw values. And then we go down here to the z-scores. Any place you see that is red or dark red, so the dark red there, for example, is now 3 z-scores above the norm for that person's age. Okay? Anything above 2 is significant, anything at 3 is very significant. So we can see these problems in beta activity from about 12 through 18, and then again from about 20 to 24 hertz over the right central parietal areas of the brain. That's where we saw in our spectral graph that we looked at here, that's where we saw the increases of beta that we talked about and those regions of the brain. And that's where we saw our connectivity problems over that same area of the brain as well. So everything lines up so we know really where the problems are. And now we know the frequency the problems are in terms of power, so we can use those as our inhibits our amplitude inhibits in our neurofeedback training. And we use the connectivity metrics for our reward band when we do our coherence training. So we're always then trying to enhance coherence where it's low, and we're then trying to inhibit amplitude where there's too much of it, which is how the therapy works. Our last step is to look at the source localization through something called S-Loretta in the frequencies that we found to be significant. So here you can see from 16 to 18 hertz. It says that right up top here. And then this shows the area of the brain exactly that's impacted, which is the precentral gyrus of the frontal lobes, which is the very back portion of the frontal lobes. This shows you some other pictures of it and the areas affected. And then from 22 to 24 hertz, again up over the frontal lobes, you can see that peak activity and where it's coming from right up there. So if we were designing their neurofeedback protocol, then we would want to incorporate those middle portions of the frontal lobes and combine them. So that will be across this area in here, if you can see that little cursor, and combine that with areas in the parietal and temporal lobes over here. So the protocol would basically cover those areas. It would be encouraging increases of coherence and trying to decrease the beta activity we saw in those analyses. So that's how we look at this. Anyone have any questions?
Will the values of the path length always be smaller than, the radius be smaller than the diameter, or is it independent of length, just the efficiency?
They are almost always, diameter is almost always larger than radius because they're further apart. Mm-hmm. And just based on the physics of the engineering, they'll be less efficient because of that. And it seems that in addition to that, most people with the types of problems we see have more problems with long range than short range connectivity. We found that in our study of people with developmental trauma. I think we probably see that in head injury and autism and other problems similar to that as well.
Great. Well, thank you so much.